Good day, and welcome to the SNN Network Summer Virtual Event. Our next presenting company is GbSciences Incorporation. It is now my pleasure to turn the floor over to your host, Dr. Andrea Small-Howard, President and Chief Science Officer. Ma'am, the floor is yours.
Thank you very much, and thank you for this opportunity to tell you all a little bit more about GbSciences and our plant-inspired pharmaceuticals. We say plant-inspired because we believe a lot of our discovery work uses the actual plants, but our development programs are very sort of pharma-friendly. So GbSciences, we're on the OTCQB, GBLX. We're a plant-inspired biotech company developing combination drugs for the prescription drug market. Our discovery program, as I mentioned, has a very unique mashup of cutting-edge AI-enabled drug discovery, as well as going back to looking at natural products research. And we believe that combination is very powerful for creating unique active pharmaceutical ingredients that are complex mixtures. Our development program takes these plant-inspired mixtures forward to the prescription drug market. One of the things that we do in the transition from discovery to development is we are using synthetic homologs of the plant-based compounds because they're more pharma-friendly and because we can make them at scale under CGMP. In addition, we are formulating in human-friendly formats. All of our formats are oral delivery formulations at present. And we have an exciting drug development pipeline that I'm going to tell you more about today. We have 4 programs that are in the preclinical stage of development, which means that we're right at that transition where we're pushing hard to get our Parkinson's program will probably be the first into the clinic, and that's both very valuable for patients who really could use some novel Parkinson's therapeutic. It's also a great inflection for our company because in general, traditionally speaking, the valuation of companies in that transition from preclinical to clinical usually translates to at least a 20-fold increase in value depending upon the situation. So this is a really exciting time to be talking about our drug development pipeline because we have these 4 preclinical programs that I'm going to mention a little bit more about today. So we're very excited. We just received positive proof of concept results from our study with Michigan State University on our COVID-related cytokine release syndrome products. And I'm going to tell you more about that in a moment. In addition, we do have a Parkinson's program where we have positive animal data and the patents are issued. We have a chronic pain program that I'm going to tell you a little bit more about, also in animal stage, and the patent has been issued. And we have an exciting inflammatory program where the patent is issued and it's proof of concept ready. Oops, okay. All right, so at this point, prior to telling you about our programs, I'm just going to give you just a high-level overview of what we believe our real strategic advantages are. And those are both, we believe we have a strategic advantage in our unique approach in bringing complex mixtures that demonstrate synergy, meaning the ingredients work better together than they do when they're used separately. And in addition, we have developed some great tools to accelerate our R&D program. Our first tool is our PHAROST drug discovery program. And it is an AI-enabled platform that has been trained to discover proprietary plant-based therapies based on traditional medicine systems. However, our proprietary therapies are not identical to anything that exists in any of the traditional medical systems today. What we're actually doing is taking the essence of some of these therapeutics and we're distilling them down to minimum essential mixtures, meaning rather than being plant extracts, we're looking at discrete, simple combinations of ingredients that can be manufactured at scale. And that also demonstrate properties that they're better than individual single drugs. Because certainly the idea of going to traditional medicine or going to plants as the idea behind discovery isn't new. But what is new is looking for mixtures instead of single ingredients like pharma has done. So the other thing that PHAROST allows us to do is it pre-validates the efficacy of our drugs by comparing across the thousands of years of history of traditional medicine. And it's able to create something that we're calling transcultural medicines, meaning we're able to look at the potential efficacy of ingredients from all over the globe that are not constrained by geography or culture, unlike the way that most of the traditional medical systems developed. We believe that these formulations will be de-risked for pharmaceuticals because of our ability to utilize this 1,000-year history in the traditional medical databases, as well as using our cutting-edge techniques for pharmacology and other aspects that will make these potential therapeutics better candidates than if we had done sort of the more traditional Western way of drug discovery. After we've identified our putative mixtures, we then use high-throughput cell models, which are very sort of out of the pharma playbook. These are just some examples of those ones that we've used in the past, but this allows us to generate tons of data that we then feed back into PHAROST, our drug discovery engine, and it helps to enrich the system. From there, we're also using another high-throughput model, the zebrafish, and this is a model for drug discovery that's gaining in popularity due to its rapid nature and its low cost. It really allows you to generate very rich data sets fast and cheap. And that's super important if you're trying to get ahead in the drug discovery and drug development game. In addition, the animal does have a 94% homology to human genes, and so it really is a great emerging model for drug development. And taken together, we've developed quite a set of intellectual property assets. We have both patents filed on our PHAROST drug discovery platform, as well as on the pharmaceutical-based formulations, which, as I mentioned before, are plant-inspired, optimized therapeutic mixtures. We have patented these around the novel API mixtures that are composed of either the natural or the synthetic homologs of these plant-derived ingredients. We've filed composition of matter and field of use claims, and we're using combinations of novel API and delivery to further strengthen our intellectual property claims. Our current portfolio contains 5 U.S. and 3 international patents that are issued on our optimized therapeutic mixtures for Parkinson's, for pain, for anti-inflammatory, and for heart disease. And as well as we have 19 U.S. and 40 international patents pending for clinical indications that have a great and broad variety of clinical indications. In fact, we've counted there's over 60 different clinical indications in that portfolio. So it's a very rich portfolio for addressing unmet needs using plant-inspired formulations. So as I mentioned, we're very excited to report that we have positive proof of concept data from our study with Michigan State on the potential efficacy of our cytokine release syndrome therapeutics for COVID patients. And we originally had been working on this project with respect to, we were looking at the immune system's response to the HIV virus, but we were able to leverage a 3-year study on how cannabinoids interact with the immune system in the face of viruses in the development of our current program. And one of the things that happened early in this pandemic, we realized that there was a need for selective anti-inflammatory treatment more than ever before. And when I first heard about the pandemic, I thought about my mom, who has rheumatoid arthritis and is on prednisone, which is one of those anti-inflammatory drugs that really leaves you immune compromised. And so I was thinking, what we really need is a selective anti-inflammatory treatment that will suppress those cytokines that are causing the most harm to COVID patients that are on the ventilators, while preserving the antiviral immunity in your body, because you want to be able to both fight the COVID virus, as well as if you're in the hospital, there's a high likelihood that you'll be exposed to other potential viral pathogens, and you need that antiviral immunity. So this led us down the path of asking, can cannabinoid mixtures suppress COVID-related inflammation while preserving antiviral immunity? So to address this, working with Michigan State, we were using what is considered a cutting-edge method of addressing human immune diseases. where you basically perform a co-culture of all the human immune cells that are harvested directly from human donors, and they're growing in dishes. We set them up in 4 treatment groups: those that were untreated, those that had an inflammatory stimulus that was virus or bacteria to mimic, and this inflammatory stimulus has been proven time and time and again to give a really strong hyperinflammatory response. We also have a positive control, which is our hyperinflammatory stimulus, plus the liquid that we mix the cannabinoid mixture in, which has a tiny bit of alcohol in there. So just to make sure that, they call that the vehicle, just to make sure that the liquid that we mixed the cannabinoid mixture in didn't have any effect on the inflammatory stimulus. The positive controls for these experiments had both the inflammatory stimulus plus the liquid that our cannabinoid mixtures are made in. And then we set up and we tested 24 different cannabinoid mixtures. We pre-exposed the cells to the cannabinoid mixtures and that for 30 minutes, and then we added the inflammatory stimulus. And then we measured the inflammatory cytokines that are associated with adverse reactions to the COVID-19 syndrome, as well as other inflammatory markers that would allow us to assess whether these mixtures were appropriate for turning down COVID-related cytokines while preserving your antiviral immunity. And we are very excited to report that all 24 of the mixtures that were derived from both our wet lab experiments with cells and our AI-enabled drug discovery software, all 24 mixtures achieved statistically significant changes in the levels of the cytokines and inflammatory markers that we were measuring. So we took these 24 positive results and we put them into categories as follows. So as I mentioned, all of these mixtures achieved statistically significance, but when we started to look at some of the cannabinoid mixtures did demonstrate the selectivity that we had originally hoped for, meaning they were downregulating those key COVID-related cytokines, but they were still preserving the antiviral immune responses. And then others, in addition to decreasing the key cytokines and preserving the antiviral immune responses, they were specifically targeting single immune cell types that are very valuable for therapeutic applications. So these first 10 were very valuable to us and were really what we were looking for. In addition, we ended up with 14 other mixtures that have great clinical potential for unmet needs. They're more broad in their approach in that they broadly are anti-inflammatory, but in general, but all of them show some promise for the clinic. So what we're doing next is we've selected the COVID CRS mixtures, those that seem selective for those properties that a lot of promise as well. We have the potential in this category to be a first-in-class drug for Parkinson, in that we've looked at the competitive landscape, and there doesn't seem to be anything with the same kind of mechanisms that we're using, so that's always a great thing. This program is also the furthest ahead. It's our lead program, and this also gives us an example of the demonstration of how our resources work together to quickly get from a potential of over 10,000 mixtures down to 3 that are in rodent testing now. And so we start with metabolomic profiles and our AI-enabled drug discovery engine. We use our discovery funnel to get down to around 1,000 combinations that were tested at the cell level. And then we go down to the medium throughput animal level, which is the zebrafish experiments. And now we're headed into mice, and then we're also developing our clinical prototypes. So one of the things that we learned early on, and these are results from our cell-based studies. People ask us, why would you bother to do mixtures? Mixtures seem like they're more of a hassle, and that's not what pharma usually does. So why would you do that? So this slide right here is a great example of why we decided we wanted to go ahead and go for the mixtures. In this slide, we're looking at the effects of the individual ingredients on the left and the effects of the mixtures on the right. And the real surprise of this experiment was not just that the individual ingredients did not perform as well as the mixtures. The real surprise for us in these early days was that conventional wisdom at the time had told us that THC, CBD, and CBN, which are sort of the major cannabinoids, that they would probably be the most important things in the mixtures. What we found even in these early days is that you could get really robust signals and not necessarily have something that people would consider a quote-unquote major cannabinoid. And so from the cell-based mixtures, as I mentioned before, we went into a zebrafish model of Parkinson's, and we achieved statistically significant reduction in the overall movement level. So we moved that back to the baseline. We reduced the resting tremor, and we maintained a normal startle response, so they weren't having adverse reactions to the drugs. We also tested multiple therapeutic mixtures for Parkinson's disease in safety and tox, and they all came out clean. It's also that we achieved our proof of concept for acute systematic relief. We're currently doing some mechanism of action studies that are suggesting that the mechanisms are neuroprotectant and anti-inflammatory, and we're using this animal data to support an IND application to the US FDA and Health Canada. So we are currently also creating our clinical prototypes, and we are working with Catalent Pharma to put our unique mixtures into their Zydis orally disintegrating tablets. We believe that this unique freeze-dried oral solid dosage solution, which is a great match for the Parkinson's patients. It's convenient, and one of the things that's known is that greater than half of Parkinson's patients have problems with swallowing. So this is great for the patients. You just put it on the tongue, and it takes typically less than 3 seconds for it to dissolve. The potential clinical advantages include improved bioavailability, increased compliance. If the drugs can't get it into the patient, then you aren't going to have great efficacy, and a rapid onset through buccal sublingual absorption, which avoids first-pass metabolism. So what are the next steps here? Well, we're doing a dose-response study in rodents at the University of Lethbridge in the fall, and we're also concurrently creating the clinical prototypes of the Parkinson's mixtures in the Zydis ODT with Catalent Pharma. After that, there'll be some talks in ADME, engage our CRO, get the pre-IND application to the US FDA, probably have a first meeting with the US FDA because we are doing a combination drug, and we're anticipating filing that pre-IND Q3 of next year if everything goes on as scheduled. The last program that I'd like to tell you about is our chronic pain program, and that's another one that we're really excited about. The idea behind our chronic pain strategy is that we are using plant-based mixtures from these are all from the cannabis plants that target the TRP family of receptors. These family of receptors are involved in the sensing of pain, and there's also some that are involved that go directly to the brain and do the perception of pain. And through both our computational analysis and wet lab experiments, we believe that having mixtures that address all of these pain-sensing receptors simultaneously will allow for greater net effectiveness in pain relief. Our mixtures are potentially first-in-class drugs. The only thing there's nothing on the market like this. Right now on the market for pain, we have your typical NSAIDs, opioids, and recommitted tricyclids and anti-seizure meds. But there's only one other product that's somewhat similar. There is a capsaicin-based pain therapeutic in a phase 3 trial by Centrexion. That's the only one that's somewhat similar, but we feel that ours is superior in that we're addressing not just the TRPV1 inputs for pain, but all of those inputs for pain simultaneously. There's also another phase 1 program by Zolira, which is an Australian company, and they have a product that's addressing the cannabinoid receptors, not the TRP receptors. That's the only other thing that are even somewhat similar to what we're doing. And so this next slide is a real demonstration of why we really got excited about this program. We have, in a rodent model, we have a proof of concept that we're combining our combinations of our plant-inspired optimized therapeutic mixtures in extended relief nanoparticles. And this is a proof of concept for this administration. So Here what you see is that in a rodent model, you could, if you gave a single oral dose of the nanoparticles that have cannabinoids in it, you got over 11 days of continuous relief with a single dose. Whereas if you took the free cannabinoids, not in the nanoparticles, you only got less than a day. And really the peak effectiveness was right around 3 to 8 hours. So we believe that this is a powerful combination of using the right ingredients from plants in the right delivery method to change the way that pain and chronic pain is treated. And we think this is going to be a real game changer. So this is just a little slide, and there's a reference there that talks about how we make our nanoparticles. Right now, we are testing the compounds separately in nanoparticles and not in nanoparticles, as well as we're testing our complex mixtures within the nanoparticles compared to not encapsulated in 2 different zebrafish nociceptive models, and we're deep in the thick of this animal trial, and I'll be excited when we can start to reveal those results. So in summary, so our next steps for the pain program, we have an animal proof of concept, as I mentioned, ongoing in Canada. We're doing production scale up for these nanoparticles. That will be followed by some talks in ADME and the usual pathway to get into our first in man. So to summarize, GbSciences has a robust drug development pipeline with 4 programs that are in preclinical. Our Parkinson's program is positioned probably furthest along, and it's the closest to their first in man. However, we believe that there are certain regulatory advantages to our COVID cytokine release syndrome product that might make it almost catch the Parkinson's. So we anticipate that within the next couple of years, we should have at least 2 products in first in man. If it's not the COVID program, then it's likely to be the chronic pain program, which is also has looking good in animal data. Behind that, we've got some great animal data to support our heart failure drugs, and we have patents that have been approved on our mast cell activation syndrome, Therapeutic, and so another one in inflammatory bowel disease that we're getting ready to get back in the saddle to do some more preclinical work as the others move forward in the pipeline. And then I'll give you sort of a forward-looking, none of this work would be possible without our great collaborators at Chaminade, Michigan State, the University of Athens in Greece, the University of Sevilla in Spain, the University of Cadiz in Spain, the National Research Council of Canada, which is roughly equivalent to their NIH, the University of Lethbridge in Canada, the University of Hawaii, and we have a preferred vendor relationship with PHAROST, who provides us with a lot of our synthetic APIs. And then Catalan, of course, has been helping us with the delivery solution. And just to look forward, the PHAROST, we've now using the PHAROST, we're at the point where it's providing us with results that we can start to act on. And we will begin our first animal studies on a non-cannabis-based product in the fall. We found depression, anxiety, and pain formulations from the Piper family of plants. And by looking within these plants, once again, as I mentioned, we're looking for minimum essential mixtures. We're going to begin the animal studies next quarter. And we believe that our minimum essential mixtures are better than the kava extracts as they are, crudely taken out, or based on, and it's also better than using a single ingredient therapy. Our PHAROST program also allows us to look for alternative supply chain options, which is great, especially within this family, because some of the plant species are grown just in very isolated and small amounts. It also de-risks these phytomedicines for prescription, as I mentioned before, by leveraging the power of looking in traditional medicine for the basis of how we put these ingredients together. And like I said, this is also an example of our transcultural medicine approach, integrating ingredients across geographies and culture distinct pharmacopoeias. So with that, I think we'll look and see what we have for Q&A. All right. So I'm going to be. So there was a question from the audience that says, does plant-inspired mean FDA botanical pathway approvals for your programs? Why not? And there, actually, no. Plant-inspired just means that we did our discovery work using natural products types approaches. But everything in development is using synthetic ingredients that are formulated very specifically and for which we know the mechanism. So we're going for a standard FDA approval, although albeit on a combination drug. And then there's another question here. Looking at the competitive landscape, what larger player does GB aspire to be? That's an interesting question, because if you had asked me 8 years ago when we started this process, I would have said GW Pharma. But now it's difficult to say, because we've really gone beyond thinking about how to take cannabis-based products to the FDA, and we're really thinking about how to tap into 1,000 years of traditional knowledge about how to use plants to help people, but do it in a way that is pharma-friendly. So, so that's the answer to that question. So thank you very much for your time, and I appreciate this opportunity immensely. Thank you.
Ladies and gentlemen, this does conclude GbSciences Incorporation presentation. Thank you for joining. You may now disconnect.